Hello everyone, my name is Anton Zalygin and I am here to talk about biophysics, about photosensitive proteins. The first question, is it important? Yes, of course. The Nobel Prize in Chemistry in 2012 was given for studies of G protein coupled receptors. You can say we're talking about physics, okay, biophysics, but chemistry here. But actually, nowadays it's rather hard to find physicists that is not working with chemistry and chemists that is not using physics in his studies. For example, quantum dots. Every physicist that is working with microscopy or nano instruments or nanomaterials, of course, he was working with quantum dots. But quantum dots got prize for work. Scientists that were working with quantum dots got prize, Nobel Prize, last year in chemistry too. And how also can we understand that it is important, at least in science? Every year, scientists doing more and more works and writing more and more articles in cited journals about photosensitive proteins. Actually, there is already more than 22,000 articles connected with this theme in cited journals. And if we're talking about photosensitive proteins, what is it, photosensitive protein? It is kind of protein that can change its structure or activity in response to light. They are found in many bacteria, plants, animals, and they mediate various light-dependent processes such as vision, phototropism, circadian rhythm, and optogenetics. The Nobel Prize in 2012 was given for G protein coupled receptor. Actually, these proteins are working with vision, with flowers, and with interaction with different hormones. And if we're talking about light-sensing proteins, there is huge advantage with these proteins because it's rather easy to activate them and rather easy to study these proteins because usually they've got different colors for their different conformations, and when we can easily check their kinetics and find different conformations of these proteins. Some examples of light-activated proteins. First of all, of course, it's rhodopsin, basis of our vision. A protein that has a molecule called rhodopsin bound inside it, and it is responsible for light perception, not only in our eyes, but in eyes of most part of animals. The next one is phytochrome. It is a protein that has bound molecule called bilin inside it, and it is responsible for sensing red and far red light in plants. Next one, cryptochrome. It is a protein that has a molecule of flavin bound in it, and it is responsible for sensing blue and ultraviolet light in plants and animals. It regulates circadian rhythms, phototropism, and other responses to light. The next one, rather interesting protein, is bacteriorhodopsin, a protein that has chromophore retinal bound in it, and it is responsible for pumping protons across the membrane of some bacteria. It converts light energy into chemical energy. And the next one is channel rhodopsin, a protein that has again retinal bound in it, and it is responsible for opening cation channels in the membrane of some algae. It allows light to control the electrical activity of neutrons It is widely used in optogenetics. Actually, there is a huge amount of photosensitive proteins in nature. In this table, you can see the most important and the most learned families of photosensors and chromophores that are part of their construction. We won't talk about all of them. Let's focus on the most interesting. And the first one I want to talk about is rhodopsin, the basis of our vision. It is also known as visual purple. It is a photosensitive protein that is found in the rod, in eyes, in cells of vertebrate retina. It consists of protein called opsin and a chromophore called retinal, type of vitamin A. So, how our vision is working? Light photon is falling on rod in our eye and being absorbed by rhodopsin. It immediately begins to decompose. Decomposition is a result of photoactivation of electrons in the retinal part of rhodopsin. Absorbed photon energy changes retinal from cis form to the trans form. This triggers a series of events that activates a G protein called transducin, which activates enzyme that breaks down cyclic guanosine monophosphate, or CGMP, it's called. There are sodium channels in membrane that are letting ions to move through membrane and keeping the difference in electrical potential between the inner and outer sides of membrane when the cell is in state of physiological rest. The decrease of CGMP levels cause the closure of sodium channels in the rod cell membrane, which leads to hyperpolarization and a reduction in the release of neurotransmitters. This signals to the brain that light has been detected. Then retinal is relaxing to the basic state and waiting for the next photon. On this slide, I have added a picture with better visualization of these mechanisms of opening channels in membrane. With opened channels in membranes, we've got resting potential between different sides of membranes, and it is physiological rest. And this potential average value is about minus 70 millivolts. But the study of mechanisms of our vision is not only the interest of fundamental medical and biological researchers. Also, there is practical use for it. It has many practical applications. For example, Alzheimer's disease. It is connected with imbalance in metals in our brain, and work with rhodopsin can help us to move these levels to basic states. Also, Parkinson's disease, Huntington's disease, and amyotrophic lateral sclerosis. We've lost one of the most popular scientists, physicists in the world because of this disease, Stephen Hawking. We can find many different types of rhodopsins in bacteria, microbial rhodopsins. We can find three most big groups of these rhodopsins that are opening cation and anion channels and closing these channels, light-driven ion pumps, and light-activated signaling enzyme rhodopsins. Actually, they all are based on retinal and its isomerization. The existence of controlled transmembrane potential difference of the membrane is the basis of many functions. Stimulation, occurrence of action potentials, conduction of nerve impulses, control of membrane excitability, muscle contraction, secretion of gland cells, transmembrane metabolism, perception, processing of information, and formation of control actions. So the first way of physical study of different, for example, rhodopsins is absorption spectroscopy. We should talk how it's working. When light is going through some body, some material, some of this light is being absorbed and some of this light is being emitted. The reason of this process is different interactions of light photons with atoms. If the energy of photon exactly equal to difference in energies of energy levels of atom, this photon will be absorbed. So, checking lines in absorption or, for example, emission spectra, we can analyze structure and properties of chemical properties of our material, of our molecules, and which properties are interesting for us in rhodopsins. Actually, in the merization of retinal molecule, rhodopsin is going through photocycle, so-called photocycle. And you can see different photocycles on this slide for different types of rhodopsins: vertebral rhodopsin, invertebrate rhodopsins, and bacterial rhodopsin. You can see different wavelengths for the peak of absorption spectra. And this wavelength can help us to separate different conditions of protein, different its states. In every moment of time when you are measuring sample with rhodopsin, you will see not only one its state, you will see mix of different states. And actually it amount of this of parts of this mix depends on lifetime of every this state. It helps us to study information about these proteins or about any changes that we are doing with these proteins and how these proteins will change after actions we are doing with it. As you see, there are almost same functions and almost same structure of different rhodopsins, but different spectra and different photocycles. One more very interesting way to study different chemical properties of biological another way to study different chemical properties of biological objects, but actually it's a physical method to study chemical properties, is Raman spectroscopy. There are some different discrete vibrational energy states in every molecule, and you can change this vibrational state when your molecule is interacting with light. And of course, the molecule will absorb some photons energy, and photons energy will change. You can collect photons. Of course, you should have a laser beam as your light, because energy of photons, of all photons, should be same. And after collecting scattered light, you can see a spectra, like you see on picture D. And this spectra is something like fingerprints of your molecule. And you can find the structure of your molecule, and you can find the differences between different molecules or modified and non-modified molecule using Raman scattering. It is one of the most powerful types of microscopy that is not distracting your material and not distracting your molecules, your proteins, and you can use this type of microscopy working with material in and with proteins in its natural form. So different forms of rhodopsins, different parts of its photocycle has different Raman spectra, and we can use Raman spectroscopy to separate them and to find them. And different spectra on this slide showing us how rhodopsin goes through its photocycle. As you understand, in different moments of time, there are not only one form in our sample. It is mixed on different forms, and amount of these forms of parts in our sample is changing during the time. But which information can we get from this? How can it help us? Let's take a look on this part of Raman spectra of rhodopsins. As you see, these Raman peaks can be separated on individual Gaussian bands. And every this band already is connected with a different form. And intensity of these peaks can show us the amount of different forms in our mix, in our sample. So, it all is very interesting, but why are we doing it? How can we use it? For example, we can use it in biomedical applications. Rhodopsin can be used to modulate the activity of specific neurons and neuronal circuits in various neurological disorders. For example, Alzheimer's disease, Parkinson's disease, epilepsy, and depressions. For example, by using the light-sensitive rhodopsin, researchers can stimulate or inhibit the neurons that are affected by this disease and restore their normal functions. Rhodopsin-based optogenetics, it's a technique that uses light-sensitive proteins called rhodopsins to manipulate the activity of cells that express them. The technique has many potential applications in biomedical research and therapy, such as modulating again neuronal circuits, controlling cardiac function, restoring vision, and enhancing hearing. The next way to use it is optical memory storage. Bacteriorhodopsin can switch two or even actually more different photostates that can represent two types, 0 and 1 for binary, or maybe even 0, 1, and 2. And by using laser beam, data can be written and read on a bacteriorhodopsin film. The next interesting way to use it is holography. Bacteriorhodopsin can also be used to create holograms, which are three-dimensional images formed by the interference of light beam. Bacteriorhodopsin films have high diffraction efficiency and can store multiple holograms in the same area. Next one, biosensors. Bacteriorhodopsin can be used as biosensor, which is a device that detects the presence or concentration of a biological molecule. By attaching a specific ligand to the protein, the binding of the target molecule can cause a change of the optical properties of bacteriorhodopsin, such as absorbance or fluorescence. And bacteriorhodopsin is a type of protein that can act as a solar battery by using light to pump protons and generate electricity. But here is one big problem, because bacteriorhodopsin absorbs light in not really white part of visible spectra. And we should use something like, for example, quantum dots to make absorbance of this nanomaterial wider, and already quantum dots will give the absorbed light energy to bacteriorhodopsin. Thank you for your attention. I hope biophysics will be interesting for you too and will help you in your future too.